Hello, thank you for tuning in to part 2 of the organic chemistry video series. In this video, I'll be talking about stereochemistry and giving an introduction to enantiomers, which is very important in organic chemistry. So first off, just some few definitions on isomers. So as you might be aware from your general chemistry class, isomers are typically molecules that have the same molecular formula, but they have different structures. For example, you might have heard of constitutional isomers. Another name for that is structural isomers. And they are simply isomers with the same molecular formula and different connectivity of atoms. So for example, all of these compounds right here have a molecular formula of C6H12. But as you can see, all of them are structured in different ways. Now, more importantly, we're going to be talking about stereoisomers. One example of stereoisomers that you might have seen already are geometric isomers. You might have heard of cis-trans isomers in either your biology class, biochemistry class, or just chemistry class. Cis isomers are isomers in which the same group is on the same side of the double bond. So these R groups are on the same side. You could also think of it as the H groups being on the same side. And in the trans isomers, the R groups on the opposite sides of the double bond. Or you could also think of it as being the H groups being on opposite sides of the double bonds. So this is like a brief subclass of what are called stereoisomers. So stereoisomers are a subgroup of isomers in general. And so as you can see, isomers can generally be split into constitutional or structural isomers, or stereoisomers or spatial isomers. As you can see, the cis-trans isomers we just talked about are a subclass of these stereoisomers, and more specifically, they're a subclass of what's called diastereomers, but we'll get into that in a bit. Our main focus in this video will be on enantiomers right here. So what are enantiomers? Well, enantiomers, also known as optical isomers, are molecules that, again, they have the same exact molecular formulas, and they also have the same connectivity of atoms, and the difference is that they are mirror images of each other. So here, as you can see, if this is the mirror plane, you can see that these two molecules on either side of the mirror plane are mirror images of each other because, for example, this red atom reflects onto this red atom, this blue reflects onto this blue atom, and so on. I've included an additional supplementary video to help you visualize this using a molecular model kit. Now, before we get into more details about this, we do want you to be familiar with not only bond line notation, but also wedges and dashes. So as you can see on this right image, we have these two molecules that essentially also are mirror images of each other. So if we draw a mirror plane here, you can see that again, the hydrogen atoms reflect onto each other, and the oxygen atoms reflect onto each other. So here, when you see just a plain line in the bond line structure, so like these two lines in purple, those indicate that those bonds lie in the plane of the page. Now, when you see a wedge like this, so these wedges, that means that the bond is coming out of the page. So these chlorine atoms are, you can imagine, as sticking out of the page in three-dimensional space. And so therefore, you can see how these two chlorine atoms would reflect onto each other across this mirror plane. And lastly, this dash right here indicates that the bond is going out of the page. So in this case, if you remember from the last video, if there's no atom attached to the dash, we generally assume it's a methyl group. But it always is good practice to write these in. So as you can see, if both of these are sticking behind the page, they again are mirror images of each other. So now that we know how to tell if two molecules are mirror images of each other and thus if they're enantiomers, we need to specify which molecule is which. Has a lot of applications in biochemistry, in medicine, and in just biochemical reactions. Because you might imagine that a certain mirror image form of a molecule or a certain enantiomer can interact with certain parts or certain proteins in the human body, but the other enantiomer might not be able to. And so you'd want to be able to distinguish between the two enantiomers. And the way we distinguish them is by having a nomenclature that designates each of these enantiomers as R or S configuration enantiomers. And the specific nomenclature or classification system is based on the Kahn-Ingold-Prelog naming system. The exact name is not necessary as long as you know how to distinguish between R and S enantiomers. So how do we distinguish between these enantiomers? Well, first of all, we're going to have to essentially assign a priority to each of the groups attached to the chiral carbon. So quickly, just to define, just as a definition, chiral carbons are carbons that are attached to 4 different groups of atoms. So in each of these molecules, the carbon in red is a chiral carbon since it is attached to 4 different groups. To tell whether each of these enantiomers is an S or R enantiomer, in this case the one on the left is S-alanine, the one on the right is R-alanine, and alanine is just an amino acid. We are going to have to essentially name priorities for each of these groups. So the way we assign these priorities is going to be largely based on the atomic mass of these groups. And what I mean by that is we're going to start traveling one distance away from this red carbon atom. So if we travel one distance away, the next atoms that we will see are this carbon atom right here, this nitrogen atom right here, this carbon atom right here, and then this hydrogen right here. And based on the periodic table, nitrogen has the highest atomic mass out of all of these 4 circled atoms right here. So we're going to assign this part a priority of 1. Then we want to assign the second priority. But as you can see, these carbons tie for their priority. So now it's always a good thing to draw out all functional groups if you're not super familiar with them. So if we draw out this carboxylic acid group, we'll see that instead we should have something like this, where again the red denotes the chiral carbon. We'll have a carbon here, oxygen, hydrogen. And so now if we further progress to sort of break this tiebreaker between this carbon and this carbon, we're going to see that this carbon is connected to 2 oxygen atoms. And in fact, it has actually 3 bonds to oxygen atoms due to the double bond. Whereas this carbon atom below, so this carbon right here, is only connected to 3 hydrogens. So because oxygen has a higher atomic mass than hydrogen, we assign this carbon atom a priority of 2. Lastly, by default, this carbon atom would have a priority of 3, and this hydrogen would be priority last of 4. Now, in order to determine whether this should be an R or S configuration, you must look at the order of 1, 2, and 3 priority and whether they occur clockwise or counterclockwise. In this case, 1, 2, and 3 are counterclockwise, and you always view clockwise or counterclockwise by positioning the 4th priority as a dash, so pointing into the page. So for example, if you instead had 4th priority coming out of the page, you'd have to try to rotate the molecule in your head so that the 4th priority is not at the back of the page, and then determine whether 1, 2, and 3 are clockwise or counterclockwise. So if 1, 2, and 3 are counterclockwise, then we have configuration S. In this case, we have 1, 2, 3, 4. And we have, if 4 points backwards, then we have 1, 2, and 3 being clockwise. So therefore, this is R-alanine. Now, one last thing I will cover is diastereomers. And essentially, diastereomers are molecules in which not all of the configurations are flipped. And what I mean by that is, if you think about enantiomers, in order for something to be the mirror image of each other, all of their configurations must be flipped. So for example, this S configuration right here becomes an R when you flip it across a mirror. And this R configuration becomes an S if you flip across a mirror. And same for the right-hand side. These S's become R's when you flip across a mirror. Now, diastereomers are not mirror images of each other, but they still differ in their R and S configurations. So in this example, there are actually 4 pairs of diastereomers. This diastereomer, this molecule right here, is a diastereomer of this molecule and also of this molecule. And this right-hand molecule is also a diastereomer of this molecule and this molecule. And this might take a little bit of thinking on your own, but essentially, you can try to convince yourself on your own time that when you flip across the mirror image, you always get opposite configurations for each chiral center. So these are the chiral centers in this case. If not all of the chiral centers change configuration between 2 molecules, then you know that they are diastereomers. And you can try to convince yourself of that. In fact, that they would not be mirror images. So lastly, before we end, I'm going to give you a couple practice problems. So these practice problems are called the game of diastereomers, identical or enantiomers. I will give you 3 problems. So problem 1, essentially I'm going to give you 3 molecules or 3 pairs of molecules and ask you to determine whether they are diastereomers, identical, or enantiomers. And we'll go over the answers in the next video. So the first molecule is going to be this molecule right here. Paired with this. The second pair is going to be this molecule paired with, and this should be a carbon right here, paired with this. Oh, and actually, I meant to write wedge here. And last problem is going to be this. So here we're introducing a ring. And this molecule. Okay, great. And you want to determine whether each of these pairs is a diastereomer, identical molecules, or an enantiomer. And I will go through these in the next video. So stay tuned.